Media Advisory, Friday, February 3, 202-3. New inoculation based on Ebola VSV vaccine concept. A National Institutes of Health research group with extensive experience studying Ebola virus countermeasures has successfully developed a vaccine against Sudan virus SUDV based on the licensed Ebola virus EBOV vaccine. SUDV, identified in 1976, is one of the 4 viruses known to cause human Ebola virus disease. The new vaccine VSVSUDV completely protected cynomolgus macaques against a lethal SUDV challenge. The findings were published in the journal The Lancet Microbe. SUDV is distinct from and less common than EBOV, but similarly deadly. A recent 4-month SUDV outbreak in Uganda that ended on Jan 11, 202-3 caused 142 confirmed cases and 55 deaths. No treatment or vaccine for SUDV disease is licensed, although candidates are in clinical and preclinical trials. One of these candidates is VSVSUDV, developed and tested by scientists at National Institute of Health's National Institute of Allergy and Infectious Diseases in Hamilton, Montana. The live attenuated vaccine uses genetically engineered vesicular stomatitis virus VSV, an animal virus that primarily affects cattle, to express at SUDV protein as a single-dose vaccine. The researchers developed VSVSUDV using techniques that led to a VBO, the VSVEBOV vaccine that the European Medicines Agency and the U.S. Food and Drug Administration approved in 2019 as the first vaccine for the prevention of Ebola virus disease. In the current studies, the investigators replaced the key EBOV protein in VBO with the comparable protein from SUDV. Next, the researchers tested the safety and efficacy of VSVSUDV in macaques. The study involved 11 animals, each of which had previously received the EBOV vaccine and then rested for 9 months. 6 macaques were vaccinated with VSVSUDV and 5 control animals were vaccinated with VSV-MARV, a vaccine candidate in development for Marburg virus. After 28 days, during which no animals showed adverse effects from the vaccines, they were challenged with a lethal dose of SUDV. None of the animals vaccinated with VSVSUDV showed any signs of disease, but 4 of the 5 control animals developed clinical signs of Sudan virus disease. The surviving control animal, which responded similarly to the vaccinated animals, surprised the scientists, and they planned additional studies of possible cross-protective immune responses. The fact that 4 control animals got sick demonstrates that pre-existing immunity to EBOV and VSVEBOV has limited effect on protection from SUDV. The investigators anticipate that giving people VSVSUDV in a dosage similar to that of VSVEBOV or VBO would provide rapid protective immunity to SUDV. Amarzi et al. Species-specific immunogenicity and protective efficacy of a VSV-based Sudan virus vaccine, a challenge study in macaques. The Lancet Microbe DOI/https://joy.org/10.1016/S2666-5247 Andrea Marzi, Ph.D., Chief of the Immunobiology and molecular virology unit in NIH's Laboratory of Virology is available to discuss this study. NIH conducts and supports research at National Institute of Health throughout the United States and worldwide to study the causes of infectious and immune-mediated diseases and to develop better means of preventing, diagnosing, and treating these illnesses. News releases, fact sheets, and other NIH-related materials are available on the NIH website. About the National Institutes of Health. National Institute of Health. National Institute of Health, the nation's medical research agency, includes 27 institutes and centers and is a component of the U.S. Department of Health and Human Services. National Institute of Health is the primary federal agency conducting and supporting basic, clinical, and translational medical research and is investigating the causes, treatments, and cures for both common and rare diseases. For more information about National Institute of Health and its programs, visit www.nagov. National Institute of Health, turning discovery into health registered.